myocilin for in vitro studies that have led to more detailed understanding of its structure, function, and disease pathogenesis. Dr. Lieberman has made tremendous contributions to our understanding of protein folding and trafficking, misfolding diseases, intramembrane proteolysis, as well as the development of new crystallographic tools for determining membrane protein structure. She was the recipient of the 2008 Glaucoma Research Foundation Schaefer Award and received the Pew Biomedical Scholar Award in 2010. She has also received numerous awards for outstanding teaching, mentoring, and outreach efforts. Most recently, she was awarded the College of Science Gretzinger Moving Forward Award. And in November of 2021, she was named the inaugural chair of the Kelly Sepchik File Faculty Endowment Award, a new effort at Georgia Tech designed to increase the number of women faculty in the College of Sciences. Dr. Lieberman has truly embodied a bench-to-bedside approach in her research on protein misfolding disorders and beyond. During the thick of the pandemic, Dr. Lieberman was a part of an interdisciplinary group of researchers at Georgia Tech who produced more desperately needed COVID-19 kits in their labs. It is truly our pleasure to welcome you here today. Please give a warm welcome to Dr. Raquel Lieberman, who will be presenting her talk entitled Structure and Misfolding in Myocilin-associated Glaucoma. How does a protein structure spell the difference between health and disease?
Thank you so much. Can everyone hear me okay? I don't know if there's like a thumbs up. Somebody can go back to the podium and give me a thumbs up. That's
everything is good.
Thumbs up. Okay, good. That was a beautiful introduction. I feel like I should send that to my mother or something. So thank you everyone for, for joining me today for my seminar. I see a number of folks that I know virtually as well as in person, and it's just an honor to be part of this series. So as advertised, I'm going to be talking about myocilin-associated glaucoma and how my lab has really taken like a very basic science approach to help understand the disease and come up with new potential interventions. Okay, I'm going to switch to the laser pointer before I forget. All right, so glaucoma doesn't really need much of an introduction for an ophthalmology group, but just to remind you, 4% of the population over 40 eventually gets glaucoma, which is characterized by vision loss due to optic nerve disease. And it's often due to elevated intraocular pressure that precedes the retinal degeneration. And so the story that the protein that we're gonna be talking about today resides in the trabecular meshwork, which is the anatomical structure that allows for drainage of aqueous humor, the nourishing fluids that bathe all of the anterior segment anatomical structures. And is thought to be intimately involved in maintaining intraocular pressure. And indeed, a diseased trabecular meshwork is associated with most forms of glaucoma. So this protein myocilin is actually expressed throughout your body, but it only seems to cause trouble in the eye, and then within the eye, like one small part of the eye in trabecular meshwork. And it's important, and perhaps the subject of another talk eventually that I'll maybe touch on later today, is that its function is not known. So all of what I'm going to be talking about today is how mutations in myocilin, as I'll describe in a second, tie into glaucoma. So mutations are found within one specific domain of the protein. This is a figure from one of our papers. It's a little misleading. The ULF domain, which is going to be the focus of my talk today, actually takes up about half of the sequence, but there are some other structural features that are of note, and of course structure determines its function. And so we're working to sort out how these pieces all go together. But for this talk, you just need to know that rare coding mutations in the gene encoding for myocilin that change its amino acid sequence, 90% of those are found in the ULF domain, and they are documented in families worldwide. So geneticists have traveled all over the world and found dominant inherited glaucoma causing mutations that are associated with early onset disease. So when we talk about glaucoma, that's 35-ish, 40 years and younger. And even though this has been, mycilin has been known for quite a while, it is still one of the very few disease-modifying targets that are known for glaucoma, which make it an interesting protein to study. All right, so what we know and what I will talk about today is that the mycilin-associated glaucoma is a toxic gain of function. So what's really important is that to date, it doesn't seem like there's any problem with not having mycilin at all. So there are some very large number of people walking around with truncation mutations in myocilin that don't produce a protein, and those individuals don't seem to have glaucoma or other major illness. But rather, when you introduce a non-synonymous mutation that changes the amino acid sequence in the ULF domain, you get a new aggregation phenotype, which is called a toxic gain of function. And I will talk a lot more in detail about this as the talk goes on. So what we know is that mycilin is predominantly a secreted protein in the cell types that we've looked at. And so it's probably participating in the extracellular matrix somehow. And so, of course, it has to be folded in the ER and then has to be trafficked out of the cell. But when you inherit a mutation and even just one copy, instead of secreting or being degraded or anything, it aggregates. And this aggregation causes ER stress and eventually kills the cells. So in this trabecular meshwork cell type, it's cytotoxic, and then you lose the cells that are presumably maintaining the trabecular meshwork extracellular matrix. And the other piece of this that makes it very interesting is that mycilin at large is not a susceptibility gene for glaucoma. And what I mean by that is that the more prevalent a mutation is found, non-synonymous mutation is found in the general population, the less likely it is to be actually causal for glaucoma. Okay, and I will come back to that as my talk as well. So what I have done here in my talk today is divided it into 3 different types of variants that you find in the olfactomedin domain. The first is the disease-associated variance. And so I'm going to be spending most of my time today talking about those. And then to complement that, I'm going to tell you some recent work that we've done on what we call common variants, variants that are found in population studies. And then I'll end by telling you about de novo variants. And what I mean by that are ones that we come up with in the lab that we think are interesting to study and probably just get us into trouble, as you'll see. Okay, so when we first started this project, we did not have a structure of any olfactomedin domain, and it did not look like any other protein in the protein data bank. And so it was possible that of the rare disease mutants that are known, that they would be clustered in one part of the protein. Well, we were sort of wrong. So this is a crystal structure that my lab published now already many years ago. And it is a 5-bladed beta propeller. And a propeller is a really cool structure. It should be your new favorite if you don't already have a favorite fold. And what's cool about it is that it kind of wraps around a central cavity. So the N and the C termini, which are the blue and red here, have to come together in this anti-parallel strand in order for the full protein to fold properly. That also means that it can't be fully formed before all of the amino acid residues are translated. Some of the other features that I'll be talking about today are central calcium ion. There's a disulfide bond that helps kind of lock the molecular clasp in place. That's what we call the N to C terminal thing, terminus. And then, much to our surprise, there are a lot of loops on the top face and the bottom face of the propeller. And in our crystal structure, they are very well ordered, suggesting that they're not really just flopping around in solution. So now that we know that it's a propeller and we know that disease variants are found all throughout the sequence of the ULF domain, well, it's not so surprising that if you sort of map the known mutations, disease-causing mutations onto the propeller, that they're all over the place. And so this is, just meant to show you that it's a pretty, it's all over. But we tried really hard in our first paper to kind of see if there were clusters, right? Because that would help us focus in on what might be important. And we came up with a few parts of the protein that seem to be important. So in between each of the propeller blades, which are these anti-parallel, 4-stranded anti-parallel sheets, there are hydrophobic side chains poking in. So changing those hydrophobic residues is going to be disruptive to the entire structure. We also noted that there's this really long loop on the top. And so disrupting that would interrupt a bunch of interactions. And then finally, there's a central calcium site, which I'll tell you more about later, that also seemed like it was, counterbalancing some negative charges coming from ligands. So we said the structure should be used to infer disease causality of newly identified mutants, right? And in 2015, that seemed like a good idea. But as you'll see, it's actually much more complicated than we ever could have predicted. Okay, but I wouldn't be standing here today if all I did was try to solve the structure, because it took us like, I don't know, like 5 years to get protein that grew crystals that diffracted that could be solved. There was a crystallographic problem that if there are structural biologists in the room, I'm happy to talk about. But we started by, making this protein and seeing what we could learn about it. And it wasn't clear from the outset that a protein that if you change one amino acid in it causes it to aggregate would ever be tractable for the kinds of experiments that we do, which require like what we call truckloads of protein, right? A lot of purified protein. So we were very glad when we finally figured out all the little tweaks that we needed in order to make purified protein to do the biophysical studies that I'm going to be showing you here. So the first experiment, and this is like ancient history here, but I'm going to be showing you some more recent data involving this technique. So I want to introduce you to what we have here. So this is a circular dichroism spectrum of a concentrated solution so that we could see what's happening in the tertiary structure. So circular dichroism is a technique that involves the difference between right and left circularly polarized light. And in the 280 to 300, we're looking at aromatics, and so it can report on the tertiary structure. So in purple and black, we have wild type and a non-disease causing SNP, our so-called negative control or positive control, depending on your mood. And then we have 3 representative variants, A420, 427T, D380A, and I499F. So these all three are representative disease variants: mild, moderate, and severe. And what you can see in the signature of the tertiary structure is that the purple and black overlay, so they're the same as wild type, and the other 3 are going to be different. Okay, so we already can tell that there's a difference between disease-causing variants and wild type for disease-causing that is not apparent in any of the neutral polymorphisms. And then that translates into a lower thermal stability. So in these experiments, what we do is we melt the protein and have a way of measuring a change in its hydrophobicity, because the hydrophobics are in the inside. And the way this is done is you report the midway point of unfolding is called the melting temperature. And what you can see here, without reading all these different variants that we tested, is that there's kind of a slippery slope. There's a pretty wide range from about 37, right, body temperature-ish, to 52, which is usually the number that we quote for wild type, and like everything in between. Okay, so depending on which mutation you have, it can be more or less destabilizing. Okay, so the least stable ULF variants, just in the isolated ULF domain, so not the full protein with all the other domains, those data really mirrored and expanded upon some very early work. And I noticed that Doug Vollrath is giving the next talk. So, I'll definitely be tuning in for this, but he was one of the pioneers really in establishing this toxic gain of function. And he did experiments where he was expressing full length myocilin in a model cell line like HEC or CHO. And he noted that D380A, which I just showed you, and a couple others, were forming this insoluble aggregate. So there were some soluble aggregates and then something that really was not soluble in Triton X100. And, that's practically, Tide where you wash our clothes. And the other little piece of it is that you can't rescue secretion by doing these same experiments in the same cell line, but lowering the growth temperature. And so in these experiments, the idea here is that lower temperature is permissible. And so translation is slowed down, everything is slowed down, you're further away from the melting temperature of a disease variant, and so you might expect some of these to recover. And then finally, an analysis that we did early on in response to a reviewer who may or may not be on this call, I don't know, is to take the age of diagnosis, glaucoma diagnosis, and compare it to the melting temperature of these variants that we had just measured. And, maybe there's a positive trend here. Take this with a grain of salt. Glaucoma's diagnosis age is not necessarily age of onset. And, myocilin, like I said, wild type, there's no place to put it on this graph either. So taken with a grain of salt, but nevertheless, there seems to be a trend that the least stable variants are the ones that cause the earliest and more severe disease. So that was gratifying. The other piece of this is that we decided to test the hypothesis that these Triton X100 insoluble aggregates were forming a special templated kind of aggregate called amyloid. Because those are the only ones from types of aggregates from my background that seemed plausible. So I could do a whole talk on this actually. So in vitro, the protein is very happy, right? We're able to make it, we can go crystals, we can take its tertiary CD, we can unfold it, we can do all these things. And actually you can incubate it at 37 degrees for about 2 weeks. And then the graduate student will say, I'm bored, nothing is happening, and stop the experiment. But if you take that same purified protein and instead of just sitting it at 37, you gently rock it back and forth, then within 2 days, you get this very characteristic sigmoidal growth pattern of a species that lights up, fluoresces at 485 nanometers. And that's a dye called thioflavin T that is known in the amyloid field to be indicative, although not like a litmus test for amyloid. And then if you take the endpoint aggregates and you look at them by AFM or TEM or your favorite imaging, you see these ropes, these long ropes. And so that gave us this hint that the aggregates were special kinds of aggregates. And I should say we've done some work on the N-terminal domain. It doesn't do any of this. And we've gone further and looked at the structure to see how it might be different and which parts of the protein might be templating the amyloid. At least in silico, we can come up with a plausible model for how the aggregates are arranged. And actually, this is also solid state NMR peptide work that comes together. So in vitro, it seems like there's an inherent propensity for the ULF domain to misfold, even without any mutations, right? You have to work kind of hard at it, right? You got to rock it, you got to agitate it, maybe there's extra aeration, right? But you can get these to aggregate, and we can have this happen with disease mutants. So there you don't have to aggregate at all. You can just incubate it at 37 and it'll form this amyloid and it'll be faster actually. But since I know this audience is more on the ophthalmology side, I thought I would spend some time showing you some images, some recent work and some old work showing that this happens in cells. So here's the HEC model. I saw Slava's on the call. So thank you, Slava, for sending us this model like 10 years ago. So he made an inducible HEC model. Doxycycline will induce the expression of either wild type or I477N myocilin, and you can see the wild type secretes. This is a dot plot, 3 different replicates, duplicate, analytical duplicates, triplicate biological replicates. And the I477N, which is another disease mutant, does not get secreted. Well, where is it? It is intracellular. And this is just our load control. So mutant myocilin is sequestered intracellularly. This is what everybody has already known is new here. And in fact, it's stuck in the ER. So these are just some confocal images. And what you see here, calnexin is an ER resident protein. And if you look at it along with I477N, mutant myocilin, then it lights up very obviously with the ER marker, and so it's caught in the ER. And that's fine. You can have aggregates caught in the ER, but are they amyloid? So here's some recent evidence that we have repeating actually some older experiments we did with Doug that show P370L, which is like the worst disease mutant we can come up with, that it also lights up, but this time we're looking at proteostat. And I don't have all of the images because they're kind of hard to look at all at once, but myocilin is in the ER. That's what I showed you before. That happens for P370L2. We have proteostat. Proteostat is a dye that's basically like thioflavin T, and it's pretty clear here that we've got a lot of yellow cells. So P370L is aggregating in the ER to make an amyloid, and that's in the full-length protein. Okay, so that's the biophysical basis for the toxic gain of function. And now I'd like to turn my attention to even more molecular detail on this aggregation and talk about the molecular chaperones that are not doing the job they're supposed to do. So mutant myocilin should be cleared, right? That's the normal thing that the proteostasis machinery is supposed to do. But somehow it's not doing its job. And so also in 2012, in the same set of experiments, we used siRNA with that I477 and IHEC model to knock down a variety of different chaperones, and only the ER resident chaperone GRP94 did anything to the accumulation of protein, the intracellular protein that wasn't being secreted. And this was work that we did with Chad Dickey, who unfortunately passed away. And if we looked at, okay, this is going away, is it being secreted? What's happening to it? In this box, you can see it's definitely not being secreted, right? So it's going away, right? Instead of being accumulated, it's going away. So it seems like, based on the co-immunoprecipitation experiments, that I477N is coming down with GRP94, forming some complex that can't go on into the normal degradation process. And we have a lot of experiments that support this. But I just wanted to show you one piece of evidence that tells you that actually, if you get rid of the GRP94 ULF interaction, then you can clear the protein with autophagy. And that's illustrated here, where you can add the GRP94, you lose the myocilin, and you lose the GRP94, and it goes away. But if you stop either Beclin or LAMP2, which are lysosomal proteins, then you reverse the process. So it seems like knockdown of GRP94 allows the mutant myocilin to be cleared, and we think it's via autophagy. All right, so the model so far is that we can come with siRNA for GRP94 and inhibit the interaction between GRP94 and mutant myocilin, which is forming some sort of complex and unable to be degraded. And by doing so, it enables some sort of other degradation that seems to be autophagy. All right, so mutant myocilin and GRP94 forming a complex, and that is like a dominant negative effect that prevents proper retrotranslocation to the proteasome, and that is having toxic consequences because you can't just add a whole bunch of protein to the ER and think everything will be okay. So now, instead of doing siRNA, we next try to do this pharmacologically. So GRP94 inhibitors are being developed in the lab of Brian Blagg, and he sent us his favorite compound, 4-BRBNIM, and we treated the I477N IHEC cells with increasing amounts of this compound, and sure enough, the myocilin is going down, and none of these other chaperones that might, be affected by this, dysregulating the GRP94 seem to be affected. And then we've done a lot of work in molecular to sort of tease apart like what's going on between the ULF and the GRP94. When are they interacting? How are they interacting? GRP94 is 94 kilodaltons. It's huge, and it's a dimer. So there are lots of different potential interacting points. But what we could show very reproducibly in vitro is that the presence of GRP94 accelerates the initial aggregation And so here we have our baseline. This is when we aggregate the ULF, and this time we're doing it at higher temperature, so we don't have to wait so long. And here it is with GRP94, and you can see the slope here of the red line is higher than the slope of the blue line. And we know now that that has to do with an N-terminal unstructured region of GRP94 that seems to potentiate that. But when we come in with the inhibitor of GRP94, something different is happening. At first, it seems like it's not doing anything, and maybe that has to do with how much compound we put in, but eventually it levels off, and it's clearly changing the pathway and changing something about the interaction. And again, this is thioflavin T fluorescence that we're looking at. So we're looking at the production of amyloid. But what's really clear is that when you look at the gel, what exactly is happening. So I'm going to take you through this quickly, but hopefully as clearly as possible. So we have a number of controls, and they're kind of boring, but the sample is either the soluble fraction, the wash, or the pellet. Okay, so at the end of this, we have these pellets that look like fibrils on the AFM grid. So when you have the GRP94 and the mycilin just together, you can see they both are aggregating, co-aggregating one at a time. And it does, this is 1-to-1, but we've done it 10-to-3 and other sort of more catalytic scenarios. Once you start to add the inhibitor, so it's going to be inhibiting GRP94, now in the soluble fraction, we have a lot of GRP94 and maybe a little bit of the ULF left over. Then we wash our pellet, so we're not, contaminating our results. And in the pellet, compared to when we didn't have the inhibitor, we have a lot less GRP94. So it's still aggregating because we're not, ULF is still aggregating because we're not preventing it from aggregating, but we are taking out the piece of GRP94 that seems to be recognizing and, glomming onto the aggregating protein and not letting go. So the effect of inhibiting GRP94 reduces the amount of co-precipitation at the endpoint aggregate. And presumably that allows the autophagy to take over. All right, so here's our model again. So this time we're going to be coming in with the inhibitor. So the GRP94 is still coming in and saying there's something wrong with myocilin. But when we inhibit it, GRP94 with 4-BRBNIM, now we're starting to clear the myocilin instead of having it co-aggregate. All right, so, this is kind of exciting work. If I may say so myself, it suggests that if you can inhibit GRP94, which is not necessary for this is only necessary in early stage development, that you could maybe have an eye drop that would allow the mutant myocilin to be degraded. So, we've done some SAR with Brian's lab. We are at heart, we are crystallographers, so we can solve structures with them. And we keep looking for any kind of heat shock response, which we don't see. And in experiments we did right as Chad passed away, we were looking at the mutant myocilin mouse model that exists to look whether we could replicate the mutant myocilin GRP94 interaction, and sure enough we can. And if you treat these mice, or a few of the mice, well, we just had a number of mice. If you treat the mice with the 4-BRBNIM, it looks like their IOP goes down, and it looks like it's pretty statistical back to baseline. So that's very, very exciting. All right, so I want to turn my attention now to common variants, because up until now it seems like our story ties up neatly in a bow, right? So I sort of said before, we sort of posited in 2015 we have the structure, we should be able to use our basic biochemical intuition to predict whether if you make this mutation it'll be bad or be okay, right? And we know that an incompatible residue change has these 4 features. It's non-native structure, it has compromised stability, it will aggregate at 37 without a problem, and it causes intracellular accumulation that has a lot of characteristics of amyloid. So if this works, what about those common variants? So you might remember that I said at the beginning that mycilin is not a susceptibility gene for glaucoma, meaning the more prevalent the mutation is, the less likely it is to be glaucoma causing. And conveniently, there's a website called Nomad that keeps track of variants for anyone, I guess, who spits into a cup and has their genome sequenced for whatever reason. There's no clinical data that comes in Nomad. But you can see here that there are 253 and counting missense mutations that are found in the mycilin gene. Not all of these are in ULF. Last time we checked in the ULF, it was like 100 something. So about half of them are there. But what's interesting is that even what we call common variants are not really that common, right? So, maybe you would give a threshold of like 1%. The Nomad people think sort of 1% is a common, common variant. And all of these are much less common. And if you look at the clinical significance, it's kind of all over the place. Conflicting interpretation, benign, likely, bad, uncertain significance, pathogenic, et cetera. So we thought we would test the hypothesis that there are disease variants lurking in Nomad. And the idea here is that if we can be smart and predict the mutation is bad, then, individuals who come up with mycilin mutations, maybe we would counsel them to seek, ophthalmic care earlier than, maybe they would otherwise. So we came up with a list of probably tolerated substitutions. So lysines to arginines, leucines to isoleucines, and a few others. And then we came up with incompatible substitutions, ones that we thought would be problematic. Changing a negative charge to a positive charge, or changing, polar to aromatic or hydrophobic, or just making something really, really big. So we tested the 16 most prevalent variants, which again are not really that prevalent, but they are prevalent relative to, for mycilin. And we looked for evidence of misfolding. Okay, so how did we do? Well, of the 16, only 13 we basically were all just the same as wild type. Okay, so that's for us is kind of boring, but produces a lot of data, but it all overlays. Okay, so there was wild type like secretion in our assay. The thermal stability values were all in the range of 48 to 55, where wild type is 52, and we solved 11 out of the 13 structures. So they all, as you can see, look identical to wild type. You'd be hard pressed to say which one was which. So how did we do? Well, we were right about the probably tolerated substitutions, but there are clearly some that we messed up, right? That we improperly predicted. Even ones like E to K, which again is a charge inversion. So it turns out we did 16 and we got 3 of them wrong. That's like 80%. So that's like a B minus, I guess, which isn't so bad, but it's not great. All right. So as you can see, there are 3 outliers that are not green, meaning they are not wild type like. So I want to tell you a little bit about those. So E300K, so again, that's a charge inversion, S to F, polar to aromatic, N to Y, polar to aromatic, exhibits some characteristics of glaucoma causing variants. So E300K, this one is kind of the most interesting, I think. It is not secreted, not at all. It definitely accumulates intracellularly, which I'll show you in a minute. It is destabilized and it has a wild structure. So I showed you this tertiary CD spectrum before. All of the wild type, all the ones I showed you before are down here in the gray, and this one has this huge peak around 270. That's different than anything we've ever seen before. We have no idea what's going on. And then our other 2, they have normal secretion, but they're not super stable, and they do have some intracellular accumulation. Maybe there's a change in the folding pathway. It gets kind of stuck due to the presence of phenylalanine. We're not really sure.
Let me interrupt you for a second with questions so that we are live and listening to you.
Okay, good. What time is it actually?
What cell line you're using for the secretion assay?
So the secretion assay in this case is a it's transient transfected hex cells, and that's the model that Doug introduced.
And for crystallography, are you using bacterial expression or are you using
it's bacterial expression, but there's no there are no predicted like glycosylation sites or anything like that, and E. coli seem to be sort of fine making it for us, and they would.
Thank you. So now we connected.
Okay, good. So I wanted to ask the question of whether those intracellular aggregates had that proteostat amyloid character. So here are wild type images. Sorry, I'm not showing you all the panels. This is just the overlay. This is all very recent work. And P370L, which is our positive control, that's the one I showed you before, where everything is co-localized and proteostat positive, plus other vehicle and other controls that we did. There is the P370L is clearly co-localizing with proteostat and the ER. And if we look at these three, common variants for which there's no clinical data, it also seems to be to the best of our ability to analyze it. And so there's something funky going on here, I would say. Oh, I'm sorry, I don't have the p-values, but they're very significant, right? So we've identified these common-ish variants in NOMAD. Most of them are boring. Most of them are stable. They don't aggregate. They have normal secretion, and they don't seem to be disease relevant. And all of that is in line with, geneticists telling me that myocilin is not a susceptibility gene for glaucoma. And then we have a few outliers that have clearly non-native structure. They're destabilized, and they seem to have intracellular aggregation. We don't know what they really might be doing, right? They could be new sources or types of misfolding. The cell may handle them differently, but again, these are just data from a database. There's no clinical data to know whether, there's any glaucoma or anything like that. With the remaining time, I want to talk to you about variants that we came up with from our brains, which got us into a lot of trouble, right? So we're trying to figure out whether something is pathogenic or not by being smart about it, right? And for the most part, we did okay. But here's where it gets a little bit murky. In order for me to explain what we did, I need to take a step back and tell you more about olfactomedin domains. So olfactomedin domains in general, they're only found in multicellular organisms. So the earliest, type of olf domain comes from like nematodes, all right? And that one is in a different organization, but the olf domain, like I'll show you in a second, looks identical. And here it is. Its structure is superimposable with our myocilin olf structure. You would not know the difference. And this is work that we did when we were having trouble getting the olfactomedin domain from myocilin to crystallize. But what's crazy is that instead of having a calcium site in the middle, as well as a sodium site that we're not sure what's going on, the gliomedin olf, which is the more primitive, does not bind calcium. There's a single ion in the middle. It's not calcium, it's sodium. And on top of that, it is like a rock. It does not melt until 69 degrees. And that is essentially thermostable, right? This is mesostable 52, that's okay for human protein, but this is really a rock, okay? So we naively asked the question, well, we make a bunch of mutations and they either do nothing or they destabilize. Maybe we can come up with something that will increase the stability of the olfactomedin domain. And we thought that if we did that, we would learn about evolution, like maybe did myocilin evolve from other olf domains or not? How? Is there some reason for the calcium binding to exist there? Maybe we learn about misfolding or folding, like why don't we have gliomedin in our eye instead of, a protein, you look at it funny and it aggregates and gives you glaucoma. And what if any of this is functionally relevant? All right, so set out, I gave a wonderful undergraduate student the task of making a mutation in a different position of the at the calcium binding site. And here we got something extremely interesting. Okay, so any mutations at D478S are ones that we just came up with. They are not in NOMAD and they're not disease associated. And our reference point here again is this like primitive olfactomedin with no calcium, that's a rock, and our myocilin olfactomedin domain, which is mesophilic. And then the other benchmark we have is our favorite disease-causing mutation D380A, which is thermally destabilized and does not bind calcium. And we know that this aspartate here is one of the calcium binding sites. So a normal, reasonable prediction would be that it doesn't matter which ligand you remove, you would get the same effect. And so we expected to ablate calcium binding and get about the same thermal stability decrease. But surprise, no, we did not. So we switched the aspartate to the serine that's found in gliomedin, and sure enough, we were wrong. So instead of having a protein that melts at 52, we now have one that melts at almost 59. That is a very big difference. And it doesn't bind calcium like we expected. And what's interesting is that D478, you can mix and match with your favorite disease-causing mutants. So D388 is the one I just mentioned. P370L, I said, was like the worst one we have. And Y437 was the one that the mouse model is made of. And you get basically wild type back with no calcium binding. And actually helps the secretion problem. So, you know, here are all the wild type is secreting. So again, this is triplicate of a dot plot here. P370L is intracellular, Y437H is intracellular, not secreted. D478S secretes like gangbusters. And when you mix and match it with P370L or Y437H, you can start to see some secretion happening, which we've never seen. It's a little bit less for Y437H, but it works really well for P370L. So we can rescue cellular secretion. So D478S is stable, but here is like the really mind-blowing thing, at least for a molecular person like me. So we lost calcium binding, like ionic actions, supposed to be really good, right? Supposed to be really strong, long distance. We lost that, but we gained thermal stability. And the second piece is that we would be hard pressed to tell you by the solution methods that I've introduced, the circular dichroism, which is the thermally stabilized one and which one is the disease variant. So this is our old data that I showed you very early on. And here is the new data with all our D47 variants, where wild type is the black one here and the dark red here. The D388 is the gray here and the green one here. And you know, you can't tell which is which, okay? So the non-native structure in D478S is basically the same as we would say as a signature of the disease variants by CD. And then moreover, when we solved the structure of D478S, we found many structural defects. So there's this blade that's shifted, so they're shifted over. D478S is moved out. There's helix unwinding, so we have a helix here. It's gone. We don't see it in the crystal structure. And then some of these loops are shifted over and moved over. So, you know, secondary structure is supposed to be stabilizing. We've lost some secondary structure. Ionic interactions are supposed to be stabilizing. We've lost ionic interactions, and yet the sum total of D478S is to be more stable than wild type. And so there's some connection between the calcium and the helix. You know, we're not really sure. So can we stabilize our old protein? Yeah, all we have to do is switch from our disease-causing aspartate alanine mutation to ASP D478 and either make it an alanine serine or asparagine. And so at least it seems like kind of maybe, right, the calcium binding site is actually a liability, right? If you have to create it, that means that, you know, it has to form very well and bring the negative charges together. And if it doesn't do that properly, then that's a problem. The other way we were able to do this was with computational biologists. There's a whole discipline of protein design, and we worked with computational biologists to mess around with 10% of the amino acid sequence. And here we left the calcium site intact. And again, we got a ROM, and it looks like identical to wild type. Okay, so there's a huge amount of extra stability that you can engineer into this protein. So here were the opportunities. You know, is there a reason for calcium binding? Well, we know that proteins are not designed for thermal stability. They're designed to do a job. So the calcium must be there for a reason. Misfolding, why don't we have a more stable Olf domain in the eye when we can make one very easily? Probably there's a functional reason, but we don't know what that function is. Like I said at the beginning, we don't really know what mycilin is doing when it's not causing disease. Okay, so how does a protein structure spell the difference between health and disease? Well, obviously it's very complicated. So we know the characteristics so we have told you about the glaucoma causing variants, the common variants, and then the ones we came up with ourselves. So I think that we've sort of delineated, you know, molecular characteristics of the rare disease variants. where there's non-native structure, compromised thermal stability, facile amyloid aggregation, and intracellular accumulation. And if we properly identify those, then it seems like we have a direction to go with to inhibit GRP94 as a possible therapeutic direction, because it allows the mutant myocilin to be removed. In our work on the common variants, it seems to hold water that myocilin is not a susceptibility gene. That most of them that we looked at, the vast majority were boring. They were just like wild type. But there were a few that were clearly not wild type like, and we don't know what they're doing in there. But when we look at the de novo variants, that's where things get a little tricky, because we can clearly come up with not very, innovative ways to switch up the ULF domain that will make it more stable, have a non-native structure, and we don't really have a way of predicting whether a mutation will be destabilizing, despite our best efforts to. There's a lot that is still going on that we don't understand. But from our de novo variants, I think that we've really learned that there's something about the metal site that's really important. It wouldn't be there if it didn't have to be, I think. So there's a lot still that we have to do. And I just wanted to thank the people who did this work. So the early work was done by a fantastic postdoc turned research scientist, Shannon, a great graduate student, Rebecca, other graduate student, Susan, Nikki, and then the folks in the lab now, we have Dustin and Max and Kamisha and Haley and Brett, who are, some of them are sponsored by T32 and R1, and supplements, and local fellowship. And then I've been extremely grateful for the NEI funding that has really fueled all of this work. So I thank you for your attention, and I look forward to our discussion. Next stop, share.
Let me start before going to people who have questions here. I have a question related to ER stress, what you have presented. It is reminiscent of P23H rhodopsin mutant that for many years was believed to cause such a strong ER response, but it was for transgenic rats that had a much higher expression of the protein than typically you will have it for rhodopsin. And in knock-in mice, this protein could be easily degraded. So that's kind of background of my question. You're pushing expression in your expression system as high as possible because that you can detect better, and you're selecting for transfection the highest efficiency. How closely that mimics stoichiometry in vivo?
Yeah, this is something we think about a lot. And I see Dan is on the call. It's really hard to get cells, like primary cells, TM cells that have like known mutations in the myocilin to do any experiments. And based on what we know about the system, we would expect those cells to be really sick. And TM cells are really hard to work with in general, and you can't really transfect them. And so there are all these just like fundamental questions we can't answer. So I think the IHEC model that Slava gave us is the best we can do, because at least we can turn it on and off. And when we compare like the results that we get, like for the reducible model and the what's it called, and the transient transfection, there are some differences, and we're just trying to use like as many model systems as we can to build this. But I mean, you can say the same thing about the in vitro experiments, like how low concentration do you have to go before maybe it's so diluted it doesn't aggregate. But I guess, we know that in trabecular meshwork, it's expressed at relatively high concentrations. I don't know what that number is, but, we're doing the best we can, I guess, is the bottom line. But I'm happy if there are some new sort of stories out of the, other systems that would be useful to us. I would love to hear more about, what we might be able to do.
Let's go to Manabal.
Hey, Raquel, thank you for this talk. It was great. I have a question regarding this IOP and the biochemical data that you showed when you inhibit the GRIP94. It lowers the intraocular pressure only in the myocilin mutant mice. Okay, this is the mouse story that you have with Brian Blagg and Chad Dickey. And I see that the IOP goes down significantly only in the myocilin mutant mice, whereas in the normal mice, it doesn't change anything. So how can you think like this would be a good drug for primary open angle glaucoma? It might be more for
only for disease variants.
Oh, okay.
Right? There's no reason. Well, okay, we have another paper that we're working on now that we're going to try to argue that maybe wild type should be in people's mind for general OAG and ER stress. But at the moment, there's, we can only ever hope to help, the 3 million people, if you do the math, who have these mutations. Yeah.
Okay. That's what my question was. Actually, I'm in Indiana University, and Brian is a good friend of mine. We are planning to work on this. I was thinking to get those myocilin constructs from you. I'll send you an email.
Okay.
Very good. So we have Dan Stemmer in our seminar today. He will be the final commentator of your presentation today and questions. So Dan, stay tuned. You'll be called.
Okay. Okay. No pressure, Dan.
Hi, this is Dorota, and it's as usual. It's amazing presentation. I'm here, and I have so it is well known that there is high co-occurrence of Alzheimer's and glaucoma, and I was wondering the probability is higher for patients with mutations in myocilin and those that aggregates as similarly to amyloids. Actually, and has anybody looked for myocilin aggregates in the brains of other patients?
So I don't know that anyone has specifically looked at that. I mean, these patients are hard to find. Like I asked the Emory ophthalmologist, like, how often do you come up with someone with myocilin mutation? And, it's basically never. These people are found in small villages, but, clearly throughout the world. But yeah, it would be really, really interesting to look at that. But I don't know what's known about how well these families are tracked, right? To be able to do all those kinds of experiments and to come up with a statistical, analysis.
Thank you.
Zach.
Hello, thank you so much for the really interesting talk. So I had a question kind of about the OLF domain as it relates to the rest of the myocilin protein. So functionality wise, like what do you predict the other domains, like what properties do they have, or do you expect them to serve, especially in vivo relative to the OLF domain? And have you thought about, I don't know, maybe this is published as well, but like a peptide tag labeling of secreted myocilin in mouse tissue to kind of demonstrate whether the secretion of the protein is really driving normal biological function, or if it's just, or if when the protein aggregates in the ER, you get cellular stress and death of trabecular meshwork cells.
All right,
and that's causing glaucoma. Sorry.
Let me see if I can piece that together. So Slava has made a knockout mouse. And those mice are mostly fine. They have, I think, weak hind legs, and Slava can correct me if I'm wrong, but there is no obvious ocular phenotype. So that answers maybe part of your question about like what happens if you don't have it there or it's secreted without the ULF domain. So the ULF domain is the troublemaker for sure. To answer yours, hold on, now I don't remember what the first question was, which is why.
Sorry, I was asking how the structure and the function of the OLF domain relates to the other parts of myosin.
So this is something that we're working on. So, our approach has just been to like cut it into like manageable pieces and then put it all together. So we have looked at all the structural pieces to the extent that we can right now. what we think our best understanding of the model, which by the way is terribly predicted by this new AlphaFold. I mean,
I was going to ask about that as well.
like hashtag AlphaFold fail, like major AlphaFold. So it doesn't do well, and it's because there are parts of the protein that, I mean, it does a great job with the ULF domain, but you know, I told him how to do that because it's my structure. But it does really poor job of oligomerization. So we think that at minimum there are 2 disulfide bonded dimers that are forming a tetramer, but that the tetramer is formed by non-covalent interactions. So anytime we see like weird misfolding with other disulfide bonds in the N-terminus, and that's a problem. So we think that it's sort of extending out the 4 ULF domains, so 2 pairs of ULF domains, but it's very prone to proteolysis. The role of the structured coiled coil region, I mean, it's sticky. I'll tell you, that's a huge pain in the, you know what, for us to deal with. So it could be sticking to membranes. Well, it sticks to membranes in the lab, it sticks to plastics, it sticks to lots of things. So my guess is that it sticks to something, holds on, and lets the ULF domain do something else. But what that other else is who knows? Who knows? Yeah.
Thank you.
Okay, before we go to Emma, let me check on the web. Xiaohua, are you satisfied with the answer?
No. Hi, it's Xiao from Berkeley.
Hi.
Talk, and I do have a couple of questions, but quickly is, can you elaborate, you know, this calcium binding sites, whether these binding sites affect the behavior of mycilin in the cells, you know, and also whether the change of the calcium, you know, affects the interaction between this glaucoma causing mutants with GRP94.
Wait, can you say that? So it depends, right? You can mutate out the calcium site and cause, you know,
Yeah, I know.
intracellular
Yeah.
aggregation, or it can be secreted, no problem.
Correct. So I wondering, you know, in the cells, endogenous, that a particular calcium binding site, whether the protein really has calcium in there or not, how they affect the protein behavior.
So
Yeah,
yeah,
yeah.
I mean, if somebody wants to help me with a mouse, that would
Okay.
you know. Right, there's no, like, we just came up with it, like it was an undergraduate project, undergraduate thesis, and you know, it's like, it's really interesting, but there's you know, the scientific motivation for pursuing it, right, isn't there on the clinical side for sure. It's just there as like a control for D380A, or like the opposite of D380A.
Okay, so in your culture models, so you see this intracell aggregates with you know, GRP94. Have you tested, you know, with different concentration of the calcium? You know, you can induce different levels of intracellular calcium, whether it affects the aggregation.
Were you at our group meeting yesterday? So we have a paper where we sort this out, and then, like, yeah, and I need to write it, but it'll be submitted soon at the end of the month. Yeah.
Okay, that's fair.
But you're right that the calcium site, you know, can be, and the calcium stores in the ER can totally affect, you know, what this protein, you know, how happy this protein is to fold, even in the absence of a mutation. That's what you're getting at. Yeah.
Okay, let's move to Emma.
Hi, Dr. Lieberman. The question that I have is, do you have any prediction? What is the function of myocilin?
I have a slide for that. I don't know. So half the time I think that it's mechanosensitive, and if you pull on it to kind of unwind the helix for D478S, then it releases the calcium, and so there's got to be some calcium signaling thing. And then half the rest of the time, I think, oh, this has to be like redox because we've got all these disulfide bonds and we can flip-flop them if we're not careful. And so I don't know. That's what I think. But you know, where it is, right? I don't know if it's not associating, I don't think, with like elastin or fibronectin or anything like that. It's got to have like a special function. So, or it has to be in a special part. But I don't know what that special part
Have you found it in the cytoskeleton since it's a propeller protein?
So that's also something we're thinking about pursuing. Slava published a paper like a decade ago already, probably, where he looked at the myocilin in mouse muscle cells. And he found it, he can correct me if I'm wrong because he's on this call, that it was intracellular when they differentiated. And so that's something that's pretty interesting to potentially follow up on. One of the things that we are trying to sort out are better antibodies, because if you're looking at muscle, then we have this coil part that looks like muscle, which is how myocilin got its name in the first place. And so we want to make sure that we know what we're looking at when we do those experiments. So that's a good question. Yeah.
Rafael. Hi, my name is Rafael. I'm from Chris Falczewski lab. I'm here back in the back of the room. I have two small questions. Is physiological calcium ion concentration from the aqueous humor enough to stabilize myocilin? And the second question is if you're aware of any protein-protein interactions from the extracellular matrix involving your lovely protein you study. Thank you.
Sure. So to the first question, I think extracellular calcium is pretty high. I'm blanking on the concentration. I don't know what it is specifically for TM. Maybe Dan knows. For the second question, but it gets folded with the calcium normally, right? So it's the calcium binding happens before, and actually we can't really remove it without misfolding the protein or doing this D478 mutation. So once it's there, it's not like you can add EDTA and have it come out. It's bathed in EDTA for like days during the purification, and when we do ICP OES, we get 1-to-1 interaction. What was the second part? Sorry, I'm so sorry. Can you repeat the second part of your question?
I was asking if you're aware of any protein-protein interactions which could occur in the extracellular matrix.
So there are many individual papers that have identified interacting partners, but there's no consensus. And I think if this were an easy problem, it would have been solved already. And so for some reason, it is really hard. And I would like to retire once I figure out what the protein-protein interactions are. So I want it to last like a little while, but not too long, if that makes sense. So we're just trying to devise like new reagents and other things that we have control over to try like pull down experiments again, but maybe using new methodology that hasn't been looked at before.
Hi, Dr. Lieberman. This is Tom from Chris's lab. I have a question. You showed that it was all these amyloid structures were not dissolving in hydrophobic interactions are not the only ones that are keeping this structure together. So how about reducing or maybe included or maybe very high salt together with temperature? Did you try to do it like that? And do you have any other idea? Is it possible that it's also the non-specific disulfide bond involved in this amyloid formation?
Yeah, that's a good question. So about the disulfide bond formation. So the disulfide bond, to our knowledge, once it's formed and in the absence of a reducing agent, like nothing changes. So all of our aggregation assays we do have the disulfide bond intact. We have looked at aggregation with like a little bit of peroxide, low like acid. I'm trying to think like other, like different pHs, pH 2, pH 3, pH 4.5, and we get different aggregates that don't light up with the thioflavin T if we change the pH particularly. So there's definitely some sort of ionic something or other that can affect the endpoint aggregate, but they still aggregate. But it's typically for us, we like to have salt there because the Ulf domain is more stable. When we take the salt out, that's another way that we get fibrils. And these are things that we sorted out in our lab so that like multiple people can do the same experiment and get the same result. So if we have it without salt, then we need a certain kind of glass cuvette and do an experiment that way, and we get the same fibrils as when we have salt there. So we can drive to pathologies, tweaking a little bit, what's in the solution or not. But in our regular aggregation assays, we don't have the triton there. The triton is just there because when you lyse, cells, if you use like Emperor or something, they all have like triton or something similar in them, and they, it's just what is in the pellet after you've, sonicated and pelleted out what's there. Does that answer your question?
Yes, but very quick follow-up. So how about, let's say, 4 molar salt plus 20 millimolar DTT and temperature? Have you tried something like that?
No, we have not tried that. Yeah. We've tried guanidinium and urea, and actually chemical unfolding is much better for the protein. There's something thermal is bad. Yeah.
All right, we're going to a final question by John, and then I will give you beautiful Dan.
Hello, Dr. Lieberman. My name is John. I'm a graduate student at the PhD lab. I have a few questions, a few short questions. The first question is, you've identified the myocilin as not as a susceptibility gene, but you did identify some troubling variants. And I believe you showed a plot of the age of onset with different variants. I was wondering, since it's not necessarily a susceptibility gene for developing glaucoma in late in age, have you looked into patients that potentially are younger in age that develop glaucoma and seeing the rate at which myocilin mutations are found in the younger population that develop glaucoma? And 2 is, how commonly do key players in proteostasis, how commonly do just generally key players in proteostasis appear in early age onset of glaucoma? And my third question is, my third and last question is, for the secretory, since it's a secretory protein, is there anything that induces the expression and secretion of this protein? Thank you.
Yeah. Sorry.
Oh, no, I'm done. I'm sorry.
Okay, so to answer your first question, well, first of all, I am not the person who discovered any of this work, right? It's all ophthalmologists from early, 1997 to, or younger, late 90s to early 2000s. And so, I don't say that myocilin is not a susceptibility gene for glaucoma. That's the geneticists who have, the neighbor study and all of them, they have done those analyses. So, take that with a grain of salt. And I'm a biochemist, right? I don't ever see patients. I don't have access to patients. And my understanding is that it's hard to find in, families that it's easy to track to be able to do the kinds of experiments that you were thinking about in terms of like following along and seeing how their disease progression happens. It would be great if that could work out. Sorry, now I've forgotten if I answered the first 2 or the first. And definitely, I don't remember what the third question is.
Yeah, I think you've answered the first 2. The last question was just, whether if there's anything that causes it to be expressed and secreted. Is there any factors that initiate the expression and secretion, or if it's just constitutively expressed?
That's a great question. And so I don't know 100%. What I can tell you is that if you just take primary cells and you stain them for myocilin, the media for myocilin, you have to concentrate it quite a bit before you can see it with like a typical Western blot. But if you add dexamethasone in the media, dex will induce additional expression of myocilin, but not right away. It takes a few days until it shows up. So dex, hits the glucocorticoid receptor and has, a lot of things happening after that. And eventually myocilin secretion increases. So one of the ways that the, trabecular meshwork field sort of validates their cell lines is actually through this induction with dexamethasone. But we don't know anything else that induces the expression. It's a convenient handle, I would say, for us in the field. Yeah.
So is it my turn, Chris?
Yes, it is yours.
Raquel, very nice presentation. I enjoyed it. My question is more speculative. So disease is focused on the meshwork and dysfunction, which leads to ocular hypertension. Myocilin has a widespread expression pattern, including in the ciliary body, where it's expressed at high levels. So why is the TM susceptible? A lot of the studies you've done looking at the interaction between GRP and myocilin are in cells that, you know, theoretically aren't dysfunctional. So what makes TM unique and susceptible to disease?
That's a great question. I bet you have some thoughts on that too. But I mean, my instinct is that TM cells are really just so senescent and phagocytotic, and they're pretty different, right, than ciliary body, which is like pumping out, you know, aqueous humor and has like completely different functions. So I don't know. I mean, maybe we should focus more on ciliary body expression and see, you know, what can be learned there. I don't know if those cells can be studied that easily. It's not something that I've thought of, but yeah, I mean, now that you have sent me some TM cells to work with, you know, I can see how they sort of peter out. I mean, they're kind of annoying, right? Like they're really annoying to work with, right? They just like sort of poop out and you're like, okay, I guess they're done, right? You know, and so I think it's just so hard to figure out what to do.
It just seems like there's an X factor or something.
Yeah.
Yeah. Thank you.
It was excellent. Thank you very much.
Thank you.